Hello students, welcome to Biosastra Talks. I hope all of you are doing well and written the NEET exam very well. And I want to tell you that there are so many news coming from the NEET exam, about the NEET exam. Don't worry, everything will go well. And at this time, that was the one thing was different was this paper was a little bit tough. And according to the analysis, they have said that it came little bit tough. When compared to the previous papers, but it's okay. Maximum, if you have read well, you can maximum you'll get the good marks. Okay. So another good thing was maximum the students who have covered the NCERT books, they may have written very well because maximum of the questions came from the NCERT syllabus itself. Okay. And another thing is maximum questions came from genetics and plant physiology, which we actually in our channel, which we are discussed about. So I am very happy that there are some questions which came from the topics and the questions I have explained in the PYQ session, and we'll go on with that. So another thing was in this video we are going to see what were the question and answers came from the NEET 2025 paper code, that is 45, and we'll go on with some revision explanation and everything, so that you can analyze your paper. Okay. Let's get into the video. So what was the first question they have given in the NEET Botany paper code 2021 is the complex 2 of mitochondrial ETC, electron transport chain, is also known as. So here they are asking about what is the other name of mitochondrial electron transport chain. Options were cytochrome BC1, succinate dehydrogenase, cytochrome C oxidase, and NADH dehydrogenase. So normally we know that there are four complexes in the mitochondrial ETC. In that they are asking about the second name. If you see here in the second complex of the mitochondrial electron transport chain, normally complex 2, what is its role? What it will do? Some enzyme is like in the complex 2, normally the succinate is converted into fumarate with the help of FAD and FAD by showing the hydrogenation system. Okay, like dehydrogenation means succinate is dehydrogenated, which with these hydrogens were added to this FAD molecules. Okay, so this is the process which takes place in the complex 2 of mitochondrial ETC. So that's why normally main reaction is that here the conversion of succinate into fumarate with the help of succinate dehydrogenase, right? That's why this complex is also known as succinate dehydrogenase. So obviously what is the right answer here? The succinate dehydrogenase is the right answer here for the complex 2 of mitochondrial dehydrogenase. Okay, mitochondrial complex electron transport chain. Okay, so we'll move on to the next question. What is the next question from this paper is polymerase chain reaction. Here they have given the question from the polymerase chain reaction itself, PCR. They are asking in the polymerase chain reaction the DNA amplifies, means if the DNA is amplified, how many number of DNA are evolved, they are asking here. Normally we know what is the polymerase chain reaction. It includes 3 steps. Denaturation means one copy of DNA is denatured into 2 copies, and then they are annealed, means each copy will start annealing, means new strands were formed like with the help of transcription and everything, and then you can see that each strand will be formed into another new DNA. So here from one copy of DNA we are getting the 2 copies here. So if we see that polymerase chain reaction amplifies DNA, so what is the right answer? You can say here is normally 2n. Why 2n means I will tell you, in PCR each cycle amplifies like 1 copy to 2 copy, like one DNA doubles, and then these 2 DNA are also doubled, then they'll become 8 and 10. So you cannot estimate. Why? Because you cannot say how many DNA molecules are present in the sample that have taken in the test tube. Okay, so if we you cannot say that 1 DNA taken, 2 DNA taken when you are taking the DNA sample. So you cannot judge it. If you know the number you can say it, but you cannot count. No, that's why normally you can indicate it with n. N means nothing but the number of PCR cycles here. How many PCR cycles will happen? So 2n means each cycle will evaluate like 2 copies. No, so 2n means what? 2 into 2n means 2 into number of cycles, right? So if each one will evolve 2, if the number of cycles are like, for example, 2 cycles happen, means 2 into 2 is equal to 4 DNA are evolved. Okay, like that it will happen. So number of according to the number of PCR cycles, the number of copies will evolve. So what is the right answer here? 2n is the right answer here. Okay, we'll move on to the next question. What is the next question is, what are the potential drawbacks in adoption of IVF? So here they are asking about the IVF method. Means we know that in vitro fertilization is a method of involving the, like they'll inject the male sperms into the females, they'll do, they'll allow the fertilization of male and female gametes like the ovum and the sperm to form a baby like zygote in the lab itself, so in vitro. So this is the process normally. If you see here, so here they have given the question, what are the drawbacks they are asking. We know that for each process there will be some drawbacks or disadvantages. So here what was the options were: high fertility risk to the mother, expensive instruments and reagents, husband or wife necessary for being donors, less adoption of orphans, not available in India, and possibility that embryo does not survive. So these are the options they have given. In this you have to say what were the drawbacks. First one is high fertility risk to mother. You can say, but it's not that much high, so this is not the answer. Another thing is expensive instruments and reagents. Obviously for the IVF they need so much cost, so this is expensive, and this is the right one. And husband or wife necessary for being donors. It's not necessary. Why? Because sometimes they can take the sperm from the father but egg from another donor, like surrogacy, like that. So this is not a potential drawback. And less adaptation of orphans. Obviously when these IVFs were done, obviously the people will not show interest in adopting the orphans. So this is also one of the drawback because of the IVF method. And not available in India. Actually it is available in India. It is also not a drawback. And last one is possibility that the early embryo sac does not survive. Obviously sometimes it will happen. Why? Because due to the sperm or ova condition, sometimes the IVF was like broken down, or it will be failed in the first session only, in the first stage only. So this is also right answer. So here what were the options that were like what were the main drawbacks for the IVF are B, D and F. So these three are the options. So here in the answer, if you see what is the right answer is, which is containing the B, D, F only. Okay. And for every question I am giving you explanation, so that after seeing the video you can see the explanation and understand easily. Okay. So this is the another thing, BDF is the right answer here. Okay, we'll move on to the next question. What is the next question is, what is the name of the blood vessel that carries deoxygenated blood from the body to the heart in a frog? Means if you see the heart, frog heart, here you can see so many valves here, like each one is having pulmonary veins, like us only, it also have the pulmonary veins and vena cava and etc. Normally we know that heart pumps the blood from, like it will take the deoxygenated blood and it will pump the oxygenated blood to the remaining parts of the body, like in every organism it will happen. So here they are asking which blood vessel will carry the deoxygenated blood from body to the heart in a frog. So from body to heart, normally what is the options were aorta, pulmonary artery and pulmonary veins. Normally pulmonary artery and pulmonary veins are the name by itself, you can say that the blood flows from lungs to heart, right? Pulmonary means the veins or arteries that were connected between lungs and heart. So these two are not. And aorta are nothing but the trunks which were present in the heart. Okay, so what is the right option here? That is nothing but the vena cava. So vena cava is, these are the blood vessels that will carry the deoxygenated blood from different parts of the body to the heart of the frog. Okay, this is the right answer. We'll move on to the next one. So what is the next one here? Which one of the following statements refers to reductionist biologist? So this is from, they are asking normally what is a reductionist biologist means. Normally they have given the so many options. First one is physiochemical approach to study understand living organisms. physiological approach to study understand living organisms, chemical approach to study understand living organisms, behavior approach to study and understand living organisms. So normally physiological approach and like this means you can say there are so many doctors, but if you see what is a who is a reductionist biology means who will see both the physical and chemical methods or approaches to study and understand the living organisms, means he'll use both the physical process and chemical methods, like the chemical approach, like by adding chemicals or by using the chemical compounds from the body of living organisms. He'll read all the biology of the living organisms. So this is the right answer. First one is the right answer, means reductionist biologist is a person who approaches physically and chemically to study the living organisms. Okay, next question is, these are like statements, so it will take time. Like normally if you see the statement 1, here they are asking, it is about the RNA actually. So in the RNA world, RNA is considered the first genetic material. If you see it in the RNA only, okay, RNA is considered normally the first genetic material. Why? Because from the RNA only DNA came, not from the DNA RNA. So it is the first genetic material evolved to carry out essential life process in the RNA world, okay, like prokaryotes and everything. RNA acts as a genetic material and also as a catalyst for some important biochemical reactions in living organisms, obviously living systems, like in some prokaryotes, okay. So being reactive, RNA is unstable, but when you see that it is reactive to every method, that's why it is not a stable one, okay. And it is also single stranded, right, normally RNA, that's why it is also, okay, it is also not a stable one. So if you see the RNA structure, this is the single stranded structure here, and it's not a stable one. Why? Because it is very reactive. So if we see the options, they are asking about is it reactive or because of the evolution and everything, like because of the catalyst nature, is it non-reactive? But what is the option is, both the statement 1 and statement 2 are correct. In the statement 1 they have said about the RNA, and statement 2 they are asking about DNA is evolved from the RNA, right? So DNA is evolved from RNA and it shows the double stranded thing, and it will have the complementary reacts, but DNA is sometimes evolved from RNA because of the chemical modifications that will take place in the living organism. So here you can consider that both statement 1 and statement 2 are correct. So in this question you can say that option 1 is correct, okay. We'll move on to the next one. So next one is another thing, like here I am giving the star mark to the questions which were like we have covered in our PYQ sessions. That's why I'm giving you the star mark, that I am very happy that some of the questions came from my PYQ sessions, okay. Like maximum questions or the topics maximum that were related only, okay. So first one, what was the question is, it is epiphytes that are growing on a mango branch is an example of which of the following. So here in the previous videos we learned about what is commensalism and mutualism and examples also. Here they are asking which is what is the best example, if these are known as best example for what actually. If you see that normally epiphytes which will grow on a mango branch, normally these are like orchids. So this is a best example of commensalism. Commensalism means what, one species normally benefits while the other is not harmed or affected, okay. Means here this orchid is developing on the mango tree, means it is benefited and the mango tree is not harmed by this. So that's why this is known as a commensalism, okay. So option A is correct here. So commensalism is a process through which these mango branch is an example, like the epiphytes going on the mango branch, okay. If we move on to the next question, what is the next question is, from the statements given below choose the correct option. Here also you have to see the statements and everything. Here they are asking about the ribosomal structures of prokaryotes and eukaryotes in this question. First option is, if you see, the eukaryotic ribosomes are 80S and prokaryotic ribosomes are 70S. Okay, if you see the structures here, normally first see the image so that you can answer it easily. So, and in this you can see that prokaryotic ribosomes are 70S and eukaryotic ribosomes are 80S. So in that 70S also we are having 50S subunit and 30S subunit, whereas in the 80S we will have 60S and 40S. And we know that in the ribosomal structure obviously 50 and 30 means 80, but why they gave it as 70 means because between the two ribosomal units there is a place which is like both of them were mixed together. Like you can say that here some part of the larger subunit mixes with the smaller subunit. So this is covered inside. Okay, that's why this is removed. So that's why 50 plus 30 equal to 80, but some part is covered, mixed with each other. That's why it comes to 70s when we see it total. Okay, and another thing also in the 80s itself, you can see that 60s plus 40s. Okay, like that middle it is combined. That's why some part is covered. Totally it is 80s. Okay. So if you see the answers here, what were the options they have given is eukaryotes are obviously 80s and prokaryotes are 70s. This is right. Each ribosome has the two subunits. Normally we know that each ribosome will have two subunits. This is also right. The two subunits of 80s ribosome are 60s and 40s. Here you can see this is right. While that 70s are 50s and 30s. This is also right. This option is right. Okay. If we move on to the fourth option, the two subunits of 80s ribosomes are 60s and 20s. Here only you can differentiate, means it is not a it's not 20s, right? It is wrong. And that of 70s also 50s and 20s. Here they have given the 20s. There are no 20s subunits in the ribosomes. Okay, so this option is wrong. And if you move on to the last option, the two subunits of 80s are 60s and 30s they are saying. It's not 30s actually, it is 40s, right? Why? Because some of the subunit is mixed with the others, so that's why it is not 30s. And the set of 70s are 50s and 30s. This is right, but this is wrong. That's why this option is also wrong. So if you see here from the statements A, B, sorry, 1 minute. These two are wrong. Okay. So here you can say that A, B and C are right. So in this option, which one is having the A, B and C? The first one. So answer is the first one. Okay, you can simply guess like this. And another thing, next question is, which one of the following is an example of ex situ conservation? This is also from like ecology and everything. So normally ex situ conservation means normally it involves the in the ex situ conservation they'll normally protect the endangered species. Okay, endangered species from the location actually. Okay, what they'll do, they'll protect the endangered species that were removed by the part of the population, means why? Because which were going to extinct, they'll protect them in a boundaries like ex situ development. So here the endangered species are like growed from the environment, away from the environment. Okay. So this is known as ex situ, means they are coming out, they are not present along with the population. That's why ex situ conservation. Normally if you see the options, national parks are not ex situ. Why? Because the people will go and come and everything. In the national parks also will not see only endangered species, right? So this is the wrong answer. And wildlife sanctuaries means they are good for the growth of animals, but they are not for endangered species. Okay, so this is also wrong. This is not ex situ actually, in situ conservation. And if we see the zoos and botanical gardens, sometimes you can say that they are the prime examples. Why? Because they are normally away from the natural habitat. Why? Because zoos are constructed away from the city, right? So that's why the zoos and botanical gardens are the best examples for ex situ conservation. And protected areas, obviously protected areas are also not for the ex situ like endangered species. Okay, so here zoos and botanical gardens are the best places for ex situ conservation. Okay, next question. What is the next question here is, this is also we have covered. And we'll move on to the next question. What is the next question they have given is, this is also we have explained in our ecology and PYQs. That's why given the star mark. So this belongs to the statements actually. If you see here statement 1 is, the primary source of energy in an ecosystem is solar energy. Okay, normally the primary source of ecosystem is solar energy. This is the right answer. And the statement 2 is, the rate of production of organic matter during photosynthesis in ecosystem. In an ecosystem is called net primary productivity. You cannot say it is a net primary productivity. Why? Because it is known as gross primary productivity. Why? Because only the plants cannot synthesize the whole everything. Why? Because some of them will come from the decomposers also, right? So that's why it is not the net primary productivity. Means here the statement 2 is wrong actually. It is known as gross primary productivity. Okay, normally the energy, the rate of production of organic matter from the photosynthesis in an ecosystem is known as gross primary productivity. I have given it here. Yeah, see. Okay, so gross primary productivity, that's why it is not net primary productivity. This is wrong answer. So here, what is the right option? The statement 1 is correct, but the statement 2 is incorrect. Okay, so these are the 10 questions I have covered in this video. Why? Because if we move. Okay, that's it guys. So we'll move on to the next. So like that we have discussed about 10 questions from this session. No? So another 10 questions we'll discuss in the next video. Why? Because if we go on, it will become very lengthy and you will become bored. That's why I have divided the questions into 10, 10, 10, 10 like that. And if you want to if you want to know the answers from 1 to 10 with the explanation, just stay tuned with the Biosastra Talks channel. Like I have given some of the questions which came from the paper. Okay, from the paper code 45. Okay, paper code 45, you have to see the paper code also. And just stay tuned to my channel Biosastra Talks. And thank you guys. I hope all of you understood the explanation well, and if you like it, just click the like button, and if you have any doubts, comment me in the comment section. And if you want the next videos also, just watch the channel Biosastra Talks. Okay, thank you.